Muy bien. Okay, well, today we are very happy to have here a distinguished scientist, Dr. Carmen Sandi, who is a professor at the Swiss Federal Institute of Technology in Lausanne, Switzerland, where she leads the Laboratory of Behavioral Genetics. She was the director of the Brain Mind Institute and is currently the co-director of the Swiss National Center of Competence in Research Synapses, and also the founder and co-president of the Swiss Stress Network. Carmen did her PhD at the Cajal Institute in Madrid and postdocs at INSERM in Bordeaux, France, and in the Open University in the UK. In 2003, she had a visiting sabbatical position at the University of Bern and was then recruited where she works currently. Her laboratory investigates the impact and mechanisms whereby stress and anxiety affect brain and behavior, with a focus on the contribution of brain mitochondria and metabolism. Her laboratory adopts an integrative research program in rodents and humans to study the neurobiological mechanisms involved in motivated behavior and enhanced risk of developed psychopathologies in early life stress. Experimental approaches in her lab include a combination of behavioral, neuroimaging, electrophysiological, neurochemical and pharmacological, metabological, genetic and optogenetic methods in rodents, as well as translational work in humans with virtual reality, behavioral economics, experimental psychology, and neuroimaging approaches. Dr. Sandi has published over 220 research articles and received numerous awards and honors, including the Ron de Cloth Prize, the Van Kolff Chair at Radboud University, and Distinguished Visiting Scientist at the Hungarian Academy of Sciences. She was president of the European Brain and Behavior Society and is currently member of the Executive Councils of the European Molecular and Cellular Cognition Society and EBBS. She is currently the chair of the ALBA Network and the immediate past president of the Cajal Advanced Neuroscience Training Program and of the Federation of European Neuroscience Society. So Carmen kindly accepted this invitation to present her work, as you are going to see next. Her work on brain, mitochondria, and metabolism in stress, anxiety, and motivation. So thank you very much, Carmen, for this, for accepting this invitation, and it's an honor to have you today presenting your seminar.
Thank you so much.
Go ahead, please.
Thanks so much, Teresa, for the kind invitation and introduction. And it's a great pleasure for me to be here. Not such a great pleasure as being there visiting you. That would have been wonderful. And I had promised also colleagues and friends visiting Mexico after I stepped down as the FENS president because I was too busy in the previous years. But now with the pandemic, I think this is a good solution that at least we can meet through the virtual world. So thank you. I'm going to indeed talk about the work that we have been doing in the lab over the last couple of years on trying to understand how brain mitochondria contributes to I'm going to see if it's this one, contributes to brain function and behavior. And I would like also to share with you a little bit the kind of the journey that it took in order to be interested in this topic, and also in order to think in the way that we are currently thinking about what's the role of mitochondria metabolism in the brain. So as the title says, I'm going to look at the links between stress, anxiety, and motivation, because indeed the thinking for mitochondria taking or playing a key role in the neural mechanisms of behavior was mainly related to motivated behavior when we started thinking about it. And when we talk about motivated behavior, we can think of situations as the ones that we see in this slide, in which we normally would be motivated because of the motivation to achieve rewards by performing a certain amount of work. So effort is one of the measures that tell us if individuals are capable of exerting the motivated behavior, and also to escape from punishment. So we have both types of motivation, the one that is positive, that mobilizes energy in the body in order to work to obtain rewards, and that's something that we could see in many of these examples. But one could also imagine that there is a situation in which there is an aversive environment, and then we also have to produce effort in order to escape. And in this type of processes, these functions like resistance, being able to exert active coping as opposed to passive coping, to perseverate, endure, etc., are very important concepts. And this is, I think, what it's the topic that eventually our research is explaining, as we are going to see. So as you can see here, many of the examples that I give are indeed competitive. It can be physical effort, competing, or more mental or cognitive effort, also situations in social environments. And I was, I think, surprised how well you kind of capture the essence of this seminar when I saw the announcement that you put for my seminar, that was so similar to the one that normally I also have here, depicting this effort, maybe the stress that one has to endure, and how maybe the personality of anxiety, as we are going to see, counts there. So if we want to explain the neurobiology of motivated behavior, I think we can go to many excellent papers where there are particular brain halves and brain circuits that are defined. And I think this is a very valid approach that helps a lot our research currently. But we are very interested in our lab in understanding individual differences. And in fact, when we think of all these challenges and all these perhaps situations of goals and then motivated behavior, we see that individuals are very different, incredibly different in how they cope with these challenges, and also in how much effort they are willing to exert. And that's why when we wanted to explain these individual differences, we thought, how can we identify individuals a priori that would allow us to predict how much effort they will be capable to exert in motivated situations, so that we can explain these individual differences. So, of course, one approach that one could imagine is we could look at genetics, for example, and then try to identify genes that predict differences in genetics. But we know that until now, genetics are explaining a very low percentage of the variance normally for behaviors. Another way that we've been following in the lab in our animal work for many years is to characterize individuals for different behavioral traits or predispositions that are going to reflect more complex and global phenotypes, and then afterwards seeing if these allow us to predict these differences in motivated behavior, and particularly under stress or when the challenges are tough. And I'm going to cut a long story short, because we looked indeed at differences in different types of behaviors, but in particularly, we were considering that perhaps trait anxiety, or this behavioral predisposition of being anxious under unclear situations, we thought that maybe this could be helpful to help us identifying individuals that are going to have differences in motivated behavior. So when I talk about trait anxiety, I think it's important to explain a little bit, because sometimes we tend to think anxiety is negative. It's only related to anxiety disorders. And when I talk about trait anxiety, I'm more referring to the trait that it's used in psychology and personality psychology, that it's one of the elements of neuroticism, and that allow us during a kind of a continuum to characterize individuals following a Gaussian curve according to their differences in their predisposition, their temperament, in dealing with situations throughout their life, as either more calm or bold, emotionally stable, and these would be the individuals that are really low anxious. Or on the other extreme, we have individuals that are very highly anxious, that normally worry about everything, and that they are always thinking about the future that could be terrible, etc. And then in between, we have most of us that are a little bit in between and not extreme. But in any case, these type of instruments that allow us to classify individuals for trait anxiety allow us also to have a tool to have a segregation of individuals according to this, let's call it phenotype. So what we've done throughout the years in the lab is to realize that when we expose individuals to stressors, not all the individuals are equally susceptible to be affected, and so behavioral, let's say, dysfunctions. There are individuals that are more resilient, whereas others very quickly show already alterations. We don't need to go to very high levels of stress or to very chronic levels of stress that they start showing psychopathological behaviors like depressive symptoms. And over the years, we put together, I think, a number of studies in the lab and also the literature. And we came to propose that high trait anxiety, those individuals that are really towards the high end of the trait, they are vulnerable individuals on its own, so that regardless of being exposed to stress, these are individuals that are showing always early manifestations of depressive-like behaviors or other psychopathologies. So when we talk about trait anxiety and how to characterize individuals, most of the work in animals is done with this type of standard test that you can see here. One is the open field, in which it's an open arena. It can be squares here, or it can be round. And individuals or rats or mice normally explore. There is a trade-off between being cautious or being exploratory, and then understanding what's out there. And normally we see that those that avoid the center of the arena, normally these are animals that tend to be more anxious, or that we at least label as more anxious, because normally we can also cross-validate these results in other tests that are also tests for anxiety, like the elevated plasmase, for example. And this, I'm sure that you are familiar with. It's a test, it's a apparatus that has two arms. Two of the arms are open, so that when the animals are visiting, they are exposed to the environment, and two other arms that are closed. And this apparatus is elevated around half a meter from the floor. And there, what we see when we put the animals to explore this apparatus is that there is huge variation in how they explore, so that the time that they explore the open arms is an index of this type of trait anxiety, because it's a behavior that it's been validated also pharmacologically. So by using this approach, we can classify animals for anxiety predisposition. And it's something that I'm going to go back in a second, but I wanted to explain it well, because it's going to be the basics for the segregation of the animals, because we are going to look at these differences in anxiety predisposition and how they account for differences in motivated behaviors and also in the neural mechanisms. In humans, one of the instruments that is more popular in neurobiological research is this state rate anxiety inventory, that it's a questionnaire that allows us to classify also individuals according to their self-reported answers in this questionnaire. And something that I wanted to share with you also, because we are doing this translational work between animals and humans, and I'm going to show some of the work in humans, is that we are not so different. So using virtual reality in the lab, I'm not going to go into a lot of details. I just want to emphasize this particular finding. What we've been doing very simply, it's to translate this behavioral test from rodents to humans, and we've developed also a kind of elevated plasmase. But what I want to comment now here is the results in this sort of open field. So when the individuals are exposed to just an open room, an empty room, and they are told you can explore as you wish, then what we have here is the heat map of how they explore depending on their anxiety, personality, or trait anxiety. And we see that those that are low anxious normally explore the field, the room quite homogeneously, as you can see here, whereas those that are high anxious tend to explore more the borders of the room and less the middle. And when we compare these two and then extract the differences, as you can see on the right side, there is a significant difference in how much they explore the centers. So this is something that was quite surprising. In a way, it's nice to see that we have this translational correspondence between the behavior of animals and humans, but somehow it's giving us now another way of characterizing also humans, that it's not depending on the subjective responses that they can give to these self-reported questionnaires. But I go back to the story, and now my thinking and my rationale was saying, okay, we want to have a model in which we can study differences in predispositions to respond to challenges, and particularly challenges that involve an important component, a motivational component. And indeed, for motivation, there are different types of tests. Some of them reward-based are, for example, operant conditioning, and then the progressive ratio for breaking point. And we are using those in the lab. And also for me, motivated behavior is also, for example, the force swing test, in which animals are exposed to adversity and then how much they are willing to try to work in order to try to escape from adversity, etc. But when we started this project, we were particularly interested in this concept of the establishment of social hierarchies through social competition, because again, we think that this is another very important type of motivated behavior. We are all individuals, animals, as you can see here throughout the evolutionary scale. We are highly hardwired in order to try to always compete or to acquire social dominance, because this comes from the evolutionary inheritance with many different advantages, evolutionary advantages and adaptations. So this is something that indeed we are maybe primed to try to win a social competition, but of course it's very dangerous because many times throughout this evolutionary scale, the way to acquire it is through fighting, and then fighting can be quite dangerous. So there is an adaptation that it's also when we cannot win, giving up and then produce subordinate behaviors. So something that we observed in the lab when we were putting animals to fight, and I'm going to comment it soon, is that the animals were almost the same, but sometimes one wins and one loses. No, it's what does it make this difference? So what factors could be critical? And then again, trait anxiety could be critical in order to explain these differences. If the individuals are so similar, would it be that differences in trait anxiety make an impact and a difference in this capacity to compete? So we've done different type of experiments in humans to test if this is the case. Some of them are just simple questionnaires that we started with in order to see if it was worth to get into details here. And as you can see here, we have the correlation between trait anxiety in the population that we were studying and different type of characteristics, like, for example, dominance, motivation, competitiveness, how competitive the individuals are, or the self-perceived social status. And then we have the results in males and females. Males are in gray and females in Bordeaux color. And what we can see is that indeed the higher the trait anxiety, the lower the competitiveness, and also the less confident individuals are about their personal characteristics. Then we did behavioral economics experiments in collaboration with colleagues from economics here in the University of Lausanne. And in order to do this experiment, because subjects or the participants are answering the experiment and the task very calmly, we thought that perhaps we wouldn't see differences related to anxiety if we didn't bring some stress, because it's not the same to compete in an arena, for territory, for something that brings stress to the table than maybe being calmly doing these games. So we decided to classify individuals for trait anxiety as either high or low anxious. And then on top of that, we added another condition that was the 3-year social stress test in groups, which is highly stressful. And what we observe here with an economic game that tell us how self-confident individuals are for their competitive capacities is that those that are low anxious, normally under indeed control conditions, no stress, tend to be more overconfident about themselves than the high anxious, but maybe not so much. But under stress, it's as if these latent differences get expressed. And then we can see that low anxious individuals got overconfident, really very highly confident, and high anxious individuals got more underconfident. And that's something that we see here in this continuous of the trait anxiety index, that those that are low in anxiety indeed get very highly confident and the others very underconfident. It's like we refer to this like a stress tears apart and reveals these individual differences. And well, I think we were happy somehow that we had this evidence in humans, because we wanted to look at mechanisms, and at the same time, in parallel, we were doing experiments in animals that were working quite well. As I was indicating earlier to you, we put 2 males that were equal for all the characteristics, equal body size, body weight, equal history throughout their lives, everything the same. The only difference is that indeed when we characterize them for trait anxiety in the test that I described before, also in the LiDAR box, then what we could see is that high anxious, when they were competing against a low anxious animal for a new territory, we put them in a new territory, they had to compete. In most of the cases, normally in 3 out of 4 of the cases, so explaining a lot of variance, the high anxious animals lost the social competition. And then when we repeated again, this was very clear, so that just being highly anxious provides a disadvantage for the individuals when they are confronted with a new animal, particularly if it's low anxious, that they would be losing the competition. And of course, we were concerned as to whether or not this could be due to lack of interest to win or to fight. But when we looked at the bins of the different bins of the social competition in which we were measuring this social behavior, we saw that at the beginning, during the first 5 minutes, high anxious animals really are trying to win. They are really more combative if you want, but with time they lose. It's as if maybe they are computing cost-benefit analysis differently, or there are other reasons. It's difficult to know because we cannot ask them, but this is something that we see consistently. So, okay, indeed, now we have the possibility to know which individuals might be having differences under stress, under challenges. High anxiety provides us a way to identify these individuals a priori without having to expose them to challenges that, as we know, might also change the neurobiology. So if we want to understand a priori if they have differences that may predispose them to these different behavior or disadvantages or advantages, then now we have a way that would be to test them for this anxiety test, and then we can start asking questions as to why would they be different. So since we want to understand the brain regions that are engaged in this social competition, also in motivation, we wanted to know when we were doing these experiments, which brain regions would get engaged when the animals are competing. And for this, we did different types of immediate early gene analysis. And what we observed, here we have the activation from basal levels that you can see depicted as the 100%, is that several brain regions got activated. Normally we saw this in most of the animals that they got activated, and in particular we saw activation in the prefrontal cortex that we know it's important for social competition. It's been implicated in the mechanisms of social competition and establishment of social hierarchies. The amygdala, not so much, a little bit insignificant for the basolateral amygdala, but not so much. And what really caught our attention was a huge increase in the Nucleus accumbens. So this is something that we thought, okay, it's like a correlation. If Nucleus accumbens gets increased, what happens if we inactivate it? So what we did is to take then 2 animals now that are equal for all the characteristics, including trait anxiety as well, with the same anxiety levels. And then we transiently inactivated the Nucleus accumbens with an infusion of mucimol, particularly in the accumbens shell. And what we saw is that this animal had a disadvantage to win the social competition, implicating that the Nucleus accumbens is important for this type of behavior. So a couple of words about the Nucleus accumbens. As you know, it's part of the ventral striatum. It receives dopaminergic projections from the VTA, from the ventral tegmental area, and it receives glutamatergic projections from the ventral hippocampus, prefrontal cortex, basolateral amygdala, and also GABAergic projections from a number of regions. What it's important to say here is that we know very well from the literature that Nucleus accumbens is implicated in reward, learning, motivation, the vigor of responses also, together with some components of the dorsal striatum, and also effort, which is a very important part of motivation, as I indicated before. When we saw that it gets so activated in the animals that are fighting, we were happy because we were aware of working humans trying to identify the neural mechanisms of social hierarchy establishment. And this was with fMRI. They also see systematically that the ventral striatum gets activated in those cases. And also we know very well from the animal literature, also from the human one, that the Nucleus accumbens gets involved, is implicated in depressive behaviors, which are like the other kind of the coin of being highly motivated. It's normally depressive behaviors go with the reduction in sociability, in motivation to be social. Also anhedonia, anergia, the lack of energy to move, to act, etc. Low motivation, and also this learned helplessness, this lack of capacity to cope with challenges. Okay, so we decided indeed to focus on the Nucleus accumbens to try to see what could be different between high and low anxious animals. But first, we wanted to understand which cell types could be implicated in this type of behavior. And as you know, the nucleus accumbens has medium spiny neurons, which are GABAergic, although also with some glutamatergic presence in some of these neurons. So we have medium spiny neurons contain or represent around 95% of the neurons in this in the striatum, and they are labeled typically in the literature as those that contain D1, dopamine receptor 1 or dopamine receptor 2. There are other cell types like GABAergic interneurons, cholinergic interneurons, other projections, etc. So we repeated the experiments I told you before. Now we put the animals to compete. And then with double labeling, we looked at which cell types get particularly activated. And these are particularly the D1 containing neurons, as you can see here. And also in particular in the low anxious animals, that are the ones that eventually win the competition. And okay, this is implicating dopaminergic transmission and maybe dopaminergic mechanisms. But in order to establish if that was the case, what we did was to give a D1 antagonist into the nucleus accumbens, and we could see that this reduced the probability of the animal to win the social competition, so reduces social dominance. So we have now indeed high anxious animals are less capable to win the social competition, and they have a lower engagement of these medium spiny neurons, dopamine 1 expressing neurons. Why would that be? Are there differences in these neurons? When we looked at the morphology of the neurons with Golgi, then you have here on the left panel the dendritic arborization of a low anxious and a high anxious animal. And I think visually we already see that there is a big difference. So the high anxious animals have a poorer dendritic arborization that is confirmed by the shoal analysis, as you can see here. When we look at spine density, then we can also see a lower number of spines or spine density in the high anxious animals, in the dendrites of these medium spiny neurons. And this spine density correlates with the anxiety of the animals as it's expressed in the time that they spend in the open arms of the elevated plasmase. So now high anxious animals, we see that they have a poorer dendritic arborization. And then we wanted to see if this is confirmed by electrophysiology. And with patch clamp recordings, Simonia Story in the lab could indeed confirm that these animals have a lower excitatory inputs, lower number of excitatory inputs, meaning that perhaps this could be explaining why they get less activated, these neurons, in the high anxious animals when they are exposed to challenges. And I'm talking about medium spiny neurons, but we wanted to see if we can confirm for the D1 expressing neurons. So after the electrophysiology, the neurons were labeled with biosighting, and we could confirm that this dendritic retraction is there for the D1 expressing neurons, as you can see here. So now we have indeed these high anxious animals have poorer dendritic arborization, and we wanted to have maybe molecular candidates that could help us understanding why these structural differences and functional differences. So what we did was to take nucleus accumbens from high and low anxious animals and do microarrays. And what we found is that there was a big difference in the expression of genes that are regulating metabolism, and from those regulating metabolism, in particular also regulating mitochondria, structure and function, like mitochondrial respiratory chain, mitochondrial complex 1, aerobic respiration, etc., indicating that perhaps indeed we should look at mitochondria, which was a little bit surprising and new for us, and we had to take a decision as to whether or not we wanted to get into this field, because it's a complex field, but we got convinced by different type of thinking. First of all, that indeed mitochondria is important for energy production, and we are trying to understand neural system and neural circuit that it's very important for the production of energy in the body, for mobilizing resources in the body and guiding behaviors in order to endure, to keep going, to exert effort, etc. So I think there was an appealing thinking that perhaps differences in this metabolic capacity that would be able to energize the neurons, or at least to allow them to keep going, might be a good substrate to explain these differences in the motivated capacities. Of course, mitochondria have a lot of energy-related functions and also other important functions like the regulation of calcium influx or flux, and also the production of these reactive oxygen species, which have signaling functions. And these different functions, like the ATP, also calcium, of course, as you know very well, they are very important for synaptic events and also for neural oscillations, which are central for cognition. So giving support to the idea that perhaps indeed we should look at these differences that we obtained with the microarray. Another important piece of knowledge is that it's known in the literature, mainly from other cells, not so much from neurons, that mitochondria are highly responding to glucocorticoids also. As we know, glucocorticoids are the stress hormones that are produced in the adrenal glands and that are lipophilic and cross the blood-brain barrier and are capable of acting in many different cells in the brain as well. And the way of acting is by indeed crossing the membrane, activating receptors that normally we know they translocate to the nucleus and modulate gene transcription. And what is interesting here is that many genes that are under the regulation of these activated glucocorticoids are genes that are critical for mitochondrial structure and function, like, for example, mitochondrial transport machinery proteins or OXPHOS proteins or transcription factors that regulate also mitochondrial proteins. So this is one way to go. Another way that was revealed more recently is that also these glucocorticoid receptors, activated ones, can then also translocate directly to the mitochondria and affect mitochondria DNA expression and also other functions in the mitochondria. So there is a very good reason in this topic because I am bringing stress as well, and anxious individuals are also more prone to stress, that perhaps these factors are playing a role. So I have to ask now, are there differences in mitochondrial function in the nucleus accumbens between high and low anxious animals? Indeed, this was the first findings that we had and encouraged us to keep going with this project. And we repeated many, many times now. When we compare the maximal respiratory capacity, we extract the nucleus accumbens and then we do respiration ex vivo with the technique Ouroboros. And as you can see here, when we provide substrates for complex 1 or complex 2, we have an advantage of low anxious individuals to have a better respiratory capacity, like a better mitochondrial function. High anxious rats have the lower capacity, but also what is interesting is that they also have lower expression of complex 1 and complex 2 proteins when we look for protein expression. And also something that we see is that it's mainly neurons in the synaptoneurosomal fraction and not in glia when we look into that. Also, they have lower ATP and they have higher reactive oxygen species, which is telling us that maybe there are problems with the mitochondria of these animals, because normally the ROS should be in parallel with the production of ATP, but they are producing more ROS. So we then did experiments in order to ask if mitochondrial function would be causally involved. So again, we took 2 equal rats for equal anxiety, and then in one of them we injected a low dose of rotenone that inhibits complex 1 activity in the mitochondria, and this animal had now a lower probability of winning the social competition, indicating that indeed mitochondrial function in the nucleus accumbens is important for winning the competition. We look also at electrophysiologically, if that's the case, if medium spiny neurons are depending on the complex one. When we do little variations which are not toxic at all, very small variations, you can see that medium spiny neurons from the nucleus accumbens are particularly sensitive because they reduce the firing frequency, and we don't see the same sensitivity in other cholinergic interneurons, for example. And we also see sensitivity, and it's particularly in the D1 expressing medium spiny neurons, to the content of ATP. So indeed, these neurons seem to be really depending very much on the energy capacity and ATP for the production of firing or action potentials. So, okay, very good. There are differences in mitochondrial function, but would it be that they also have differences? I mean, where do they come from? Right now, it's functionality, what we observe there, also maybe expression of complexes. So we wanted to look at the image, and we did electron microscopy images to compare the structure of mitochondria in the low anxious and high anxious animals. As you can see here, they look quite different in these sections. They don't have differences in the total density, but they have differences in the shape. And normally we see that mitochondria in the high anxious animals cover a bigger area and also have a bigger coverage when we look at these slides. Something else that we looked at and that was quite interesting is the interaction or the contacts between the mitochondria and the endoplastic reticulum that you can see here in blue mitochondria and in red the endoplastic reticulum. This is a very important partnership for the many functions, particularly calcium buffering and other micromolecular processing. And we see that the high anxious animals have lower contacts between these two organelles. So the shape, differences in shape and the differences in these contacts led us to think also based in findings in the literature in other neurons and brain areas that perhaps molecules related to this fusion and particularly fusion and fission processes, but also for the interactions with these organelles within the plastic reticulum could be affected. So we looked at the number of these proteins, and as you can see here, Mitofusin 2 is lower expressed in the nucleus accumbens of high anxious rats, something that we confirm with all the possible techniques. This is with Western blot, but also with immunohistochemistry at the level of RNA with the RNA-scope. And it's something that we observe in the nucleus accumbens, and as you can see here, not in the amygdala. So, because it makes a lot of sense that the Mitofusin 2 is involved because of this involvement in the shape, in the fusion processes, and also in the interaction with the tethering with the ER. We decided to now treat high anxious animals with a virus to restore the levels of Mitofusin 2 in the nucleus accumbens in high anxious animals. And that's what we did in the experiment. We characterized animals for anxiety. We did the injection. And then 4 weeks afterwards, they were exposed to a number of behavioral tests. And then the brains were taken to look for differences. And as what I show here is that indeed the treatment restored the deficit in Mitofusin 2 and the high anxious animals. This also had impact in the capacity of the mitochondria to respirate. As you can see here, it's restored. And then we wanted to not only see the structure in 2D because it's a bit dismissive. We did these experiments with the EM to do a serial block analysis and then a 3D reconstruction so that eventually we would have in this piece of tissue all the mitochondria reconstructed. As you can see, the mitochondria have very different shapes. And what we found, I want to pass here. What we found is these 3 blocks, if you want. We have low anxious mitochondria that look a bit loose, not a blocked of mitochondria as we see in the high anxious animals. It's packed And then when mitofusin 2 is overexpressed in the high anxious, they look again much more similar to the low anxious. And this is something that we see in many different parameters that we extract, like the total surface, the volume, etc. It's restored. What it's maybe more important is that when we looked at all the other parameters that I explained before, like the morphology, for example, the dendritic arborization that I explained earlier, then we see that it's restored when we do this overexpression of mitofusin 2. The connectivity with electrophysiology is restored. At the behavioral level, the animals that were high anxious with this overexpression behave in anxiety test as low anxious, and in the social competition that they normally lost against a low anxious animal, that now they don't lose, they even win. And then in other behaviors that I don't have time to explain, they also got restored. So from this, we conclude that mitofusin 2 in the nucleus accumbens seems to be reflecting differences in anxiety when it's low is in those high anxious animals. And this has a huge impact in mitochondrial and neuronal structure and function and regulates stress coping behaviors. So we are now trying to understand specific projections, because I'm talking about medium spiny neurons in general, even if it's the D1 that we established. But now we want to understand D1 versus D2, what could be the contribution when they are projecting to different brain targets. And this is something that we are doing now with mice genetically modified and also with the use of viruses. I would tell you next time that we meet. So in order to finish, I'm going to pass this because I already commented. I think one question is, where does it come from? We are now doing experiments to test if there are genetic differences, because these Wistar rats are outbred, so we cannot exclude that there are genetic differences in the mitofusin 2 expression, for example, or maybe in other genes that regulate this circuit. But something that it's important to ask is if this can be programmed by early life stress. And this is a project now in mice that allow us to answer partially this question. These are mice that are exposed to peripubertal stress, so stressors during childhood and puberty. And when they are adults, they have a reduction in sociability, also the motivation to explore juvenile conspecific. And when we look at mitochondria respiration capacity 2 or 3 weeks, 3 months after they were stressed, as you can see here, they also have a reduction in the respiratory capacity. And this is now a project that we looked at many other different aspects, but I don't have time to go through the details, but I think it established the capacity indeed of early life stress to have a long-term impact and programming in this circuit and in this mitochondria. And one question in which we are now devoting a lot of effort also in the lab is in doing translational work in humans. In order to do this translational work, we start from tools and techniques that we can apply in animals and then in humans. And this is mainly what we are doing at the moment is spectroscopy in the brain. We've done until now proton spectroscopy, and we are now planning to do also phosphospectroscopy. And what we can do is to direct the coil to specific brain regions. We've been looking at nucleus accumbens, prefrontal cortex, and then we can measure a number of metabolites very precisely, because this is 14 Tesla. And I'm just going to give you a little bit of results here, but very quickly. What we find when we characterize animals for, these are mice, for dominant or subordinate, and we have other groups here, is because we also gave them chronic stress, but the important bars are the first one and the third one. These are animals under control conditions. We see that metabolites related to energy, phosphocreatine, total creatine, are lower in the subordinate animals than in the dominant ones, and also other metabolites related to astrocytic and other functions, like, for example, taurine. Taurine is much lower in the subordinate animals, and it's also very sensitive to chronic stress. So after chronic stress, we also see a reduction in taurine. And taurine is important for mitochondrial function, antioxidant, for complex 1 function, etc. So there is a reason to look into it. When we did experiments in humans, one of the major metabolites that varies in the content in the nucleus accumbens with trait anxiety is taurine. So that those high anxious humans, in this case, they have much lower levels of taurine. And also those humans that have low levels of energy, that are always tired, etc., they also have lower levels of phosphocreatine. And I pass this now. So that what we are trying to also do now is to get inspired if this can help us developing treatments or understanding which treatments could be useful both for anxiety and also for improving motivational capacities. And over the years now, we've tested different ones. As you see here, we have taurine in preparation, but we've also tested the L-acetylcarnitine, medium chain triglycerides, and you see here that the papers have been published, so that the results were good. And now we are about to launch a study in humans also to test some of the nutritional components. So I finish here by maybe giving a big or a quick conclusion that nucleus accumbens is very important. We know from the literature for motivated behavior, but also that it varies in individuals that have different capacities to endure, particularly under challenges. And this is individuals that are going to show differences in mitochondrial function, that these are related to differences in the structure and function in the neurons. And also early life stress can program these long-term behaviors and mitochondrial function. And this can inspire treatments that target the nucleus accumbens mitochondrial function to improve stress resilience. So our working hypothesis is, I think, quite evident from my talk, that mitochondria really have a role, metabolism probably as well, in neural coding for stress coping and motivated behaviors, and this is modulated by anxiety. And with this, I would like to acknowledge the work of the team that are covering all these different aspects with, I think, a lot of endurance and motivation. So I'm really grateful to everyone who's contributed to this work, and also our collaborators that I've been normally naming while I was presenting the different results. And here, I would like to thank you very much for your attention. This is a picture of the EPFL where we work, and the Lake Geneva that you can see here in front of here is France. This is Vienne, where the water comes from. And this Lausanne is around here, and this is our campus. Thank you very much. I will be happy to take questions.
Thank you, Carmen, for this wonderful seminar. And there are plenty of questions. I think Carmen Clapp, Dr. Carmen Clapp, can open the microphone for just to formulate her questions.
Yes, can you hear me?
Perfect.
Yes, yes, Carmen.
It was very exciting, and it raised many issues. One of them is, could you view high anxiety as survival advantage for avoiding dangerous conditions? I mean, it may not be negative. And also, the derivative of your study may result into, like in a long term, the development of a drug to change the attitude of people. And I don't know if that's ethically acceptable or adequate, because what are the right values? Is it better value to have low anxiety and a dominant phenotype in our society? So I would appreciate very much your comments. Thank you.
Thanks so much, Carmen. First, if it's adaptive or not, if it's a problem, I think like almost everything, it depends very much on the context. Because of course, all these traits have been selected throughout evolution because they've been advantageous to cope with challenges and threats for survival. And that's why we have all this diversity. So if the context is threatening, indeed, being high anxious is very adaptive, because those that don't care, don't are bold, etc., I think they die. And I think we have an example now with the COVID situation, that many people have been reporting after they got the COVID, young people or whatever, and they had not been worried before, and they maybe also dismissing the instructions of being careful, that afterwards they realize, oh, I was really in danger. I didn't imagine that this was so bad, and I misbehaved. So many of them maybe died because they were not careful. So I think indeed with traits, one cannot say one is advantageous or versus the other one, because it's the context that it's defining. But in general, I think it's good to have a little bit of everything. If one is too anxious, it's not good, because probably it's preventing the individuals from exploring or from trying new things or from bringing innovation. But yeah, I think this is my answer. I think it's the context is going to define everything. In humans, of course, we are not anymore in the jungle, and now most of the time we have a more or less defined environment, and our context doesn't maybe change so much. So many times being excessively anxious can be dangerous somehow, can prevent individual from enjoying many of the advantages of society, and that's why normally we are calling these extremes of anxiety eventually become an anxiety disorder. And regarding the development of drug, indeed, last week I was contacted spontaneously by a kind of an incubator from Paris that saw one of our papers and wanted to discuss if there would be possibilities to launch or to reinforce our studies to eventually develop drugs. And I think they were a bit disappointed that we didn't patent these results with Mitofusin 2, because that makes it now that one needs to identify another target, maybe related to how Mitofusin 2 is contributing, in fact, to these differences that we observe. But yeah, I think it's maybe the second part of the question. My answer has to go back to, because both questions are a little bit philosophical. I think the answer is it's going to depend on the context again. So for those individuals that suffer, and when the situation is unbearable or whatever, I think it's good to have something that make us feel more at ease. And maybe it's not a drug. What we are trying now is to see if nutritional supplements, so something that normally we are eating, like, for example, I like to comment this example of peanuts, because when we discovered that nicotinamide was improving mitochondrial function, because I didn't have time to present this, but we could boost mitochondrial function in the nucleus accumbens by giving an injection directly into the nucleus accumbens of a precursor of a booster of mitochondria. And then I tried to see which foods have this component, because this is eventually vitamin 3, niacin. And of course, one of the foods that have a higher concentration is peanuts. And then you could imagine that indeed, if you eat this regularly, maybe it has an impact on how mitochondria eventually is working, and has an impact or not. I think, yeah, this nutritional approach is very promising, and that's something that we saw with other studies in the lab. So I think one can go beyond the drug. Is that? Thank you. Thank you.
Yeah, Mauricio, can you formulate your questions?
Yes, thank you. Thank you very much for your very interesting seminar. I have two questions. The first is, did you evaluate redox state either in the mitochondria or in the cytosol in your system?
Yeah, at the moment we didn't do it. We are doing this now with the viral approaches that can change the color of the fluorescence depending on the redox state. But until now, we did it more with a chunk of tissue and not separating different fractions. But yeah, I would like to do that.
Yeah, and the second, did you see a glycolytic compensation in the group with lower mitochondrial activity?
We didn't look at the glycolytic pathway yet. We are now developing also some assays because we started looking also at astrocytes. That's something that I didn't mention because it's ongoing work. That indeed indicates that probably we have to look not only at the neuronal component, because we have also, when we look at the images, astrocytes are different. They are covering the blood vessels differently, and also the mitochondria is, the location of mitochondria in the astrocytes, in the end feet of the astrocytes is different. So we are doing these studies at the moment, but yeah, it's very interesting questions.
Thank you very much.
Mm-hmm. Thank you.
Now, Dr. Josefina Ricardo. Adelante, José.
Gracias. Okay, Dr. Carmen Sandi, my congratulations for your, not only for your excellent seminar, also by all your life, scientific life. It's wonderful, really. Excuse me for my English, please, but it's my feeling. Gracias. You publish about the relationship between a trait for high anxiety and depression. Mm-hmm. My question is to know if your experience or your opinion about another important anxiety disorder, the obsessive compulsive disorder. Mm-hmm. What is your opinion about the high trait for anxiety and this disorder? Is clear?
Yes, well, with anxiety, this is the thing that there are many different manifestations of anxiety, because normally I talk about general anxiety, but of course, this is the obsessive compulsive disorder is another type of manifestation, which is probably involving similar and additional circuit, because we know that then the dorsal striatum would be also very much implicated. And the problem is, I don't know if there are good models, animal models to study this. I wonder if you are aware of this. And this is something that I think it's preventing the development of knowledge in this field. But now with the tools that we can look at also metabolism in humans, this would be the type of questions I think that one could ask. To what extent, when we look at the brain of individuals with a general anxiety disorder, let's say, or differences in anxiety and the obsessive compulsive disorder, if we see similar metabolic issues. But yeah, I think high anxiety is comorbid and also risk factor for many different disorders, even for neurodegeneration, because it provides a lower threshold also to experience stress. But yeah, I don't know if this has been studied, to what extent it's a risk factor for obsessive compulsive disorder.
Thank you very much.
You're welcome.
Now, Dr. Gina Quirarte. Gina.
Yes. Do you hear me?
Yes. Hola,
Ay,
Gina.
hola, Carmen. Un gusto, un gusto. Excelente plática. Muchas gracias. And let me tell you that my group is fan of you. So we have a lot of questions, and actually, you know that we're working in the mitochondria because and there are two really nice papers that inspire us. The one with Bruce McEwan, the energetic view of stress, and the one with Fiona. And we really like those papers. So I want to say that.
Thank you. Thanks so much, Gina.
Yeah, and there is this PhD student. Well, now she's a doctor and she's in a hospital now doing diagnosis for COVID. She's in my group and she wants to ask you a question. And she wrote this to me, and she wants to know do you think that the level of anxiety reflects the stress level of the mother?
Yes, not only, because I think it can. Indeed, the mother has an impact. We did a study many years ago in which we characterized, this was animals, rats, and we characterized the parents, both the mother and the father, so that we had the levels of anxiety. And then we looked at how the offspring was reacting to stress. And then what we observed is that the level of anxiety of the mother, the trait anxiety, was explaining the degree of corticosterone release in the offspring. And this is something that indeed it might be related perhaps a little bit to the inheritance of mitochondria from the mother, but we also know that mitochondria is inherited, but that mitochondrial genes and proteins, many of them can also from the father. So I think here what we would need to study is in particular the mitochondria DNA in order to answer this question, if mitochondria could be in those cases, because at least we saw evidence for this particular aspect for corticosterone responses. I don't know if for stress in general might play a role, if it's inherited also through the mitochondria. I guess that's what your student was asking, no? Also.
Yes.
Thinking.
Yes, yes, I think so. I hope Renata got the answer. Thank you. And I have another question, which is very simple one, but in some way I think is difficult to know if there are differences, like if there are sex differences, like in your competition results. And I think this is an issue that is very important because almost all the studies in neuroscience are made in males, and a few people work in females. So first question is if the results that you present are in male, and if have you seen some differences between sex?
Thanks so much, Gina. I think no, you're totally right. We started thinking about social competition and doing all this in males because that's what we were using at the time, before everybody started being more concerned about the sex differences. Then, of course, all this brings a very interesting question because we know that males are more competitive than females. Males are also, on average, they are also less anxious. When we look also in animals, in humans, there is a difference in the levels of trait anxiety. So now I think it's very interesting to ask how does the mitochondria and also metabolism in the nucleus accumbens compares between males and females. And this is a project that I would like to launch. And I think it's very well justified because the questions are there beyond only looking at sex differences. In this case, because there are differences in both anxiety and social competition, I think it brings another interesting question beyond only the sex differences, which on its own, they are important enough to do this type of study. So yeah, we didn't, originally, we didn't look at females because we wanted to go into the mechanisms. And then, yeah, it takes a lot of time. And that's why we continue there. But now, indeed, this is, now we do all the studies in males and females, but we still don't have the answer to your question. Yeah. But I think it's critical.
Thank you. Thank you, and great to see you, Carmen.
Yeah, very nice to see you, Gina. All the best.
Now, Dr. Isabel Mendez says that thanks for a very interesting seminar. My question is, what factors do you think are down-regulating Mitofusin 2 in high anxiety subjects?
Yeah, this is a winning question. Of course, we are interested in knowing what could be the upstream mechanism. As I said, now we are looking at possible genetic differences, but I don't think the answer is just genetic differences, even if we find them, because when we look at expression levels in other brain regions, we don't always see a reduction. In many occasions, we don't see differences, like in the amygdala. In the prefrontal cortex and in the hippocampus, we see the opposite results. We see more Mitofusin 2 in the high anxious animals. So still, it might be that there are genetic differences that are expressed by the environment, and the environment might be the neurochemistry or other environmental factors at the cellular level that are different in different cell types and neural circuits, etc. But in fact, we are trying to understand where it could come from. So we are looking now at, the results that we obtain with the microarrays and looking at upstream mechanisms. For example, glucocorticoid receptor can modulate pathways towards the regulation of transcription of Mitofusin 2. And we know that these animals in the Nucleus accumbens, they have differences in the expression of the glucocorticoid receptor. High anxious animals have lower expression in the Nucleus accumbens, and also when we take the mitochondrial fraction and we look for protein expression, they also have lower receptor expression. So I always think that if an individual is highly anxious, normally it's going to be exposed to higher levels of glucocorticoids. And we know from the literature for many years that glucocorticoid receptors are very important in this area of the Nucleus accumbens mesolimbic system for the release of dopamine or for the extracellular levels of dopamine. And we know that dopamine is toxic to mitochondria, particularly complex 1, but this would be more maybe presynaptic. So I don't want to force that maybe it's dopamine that it's affecting this, but it might be other signaling mechanisms. So my hypothesis, but we didn't demonstrate it yet, is that indeed glucocorticoid receptors, maybe with additional factors that are concurrent, perhaps to excess dopamine or to dopamine function, might be upstream from these mechanisms. Also, there are other possibilities because the circuit is functioning differently. As we see, there is a dendritic retraction. It might be exposed to too much glutamatergic excitotoxicity throughout development, throughout life, and this could have also caused adaptations. So I think, yeah, right now we have a number of possible candidates, but for me, perhaps the stress pathway and glucocorticoid pathway is a possible candidate. Also because glucocorticoid receptors are regulating transcription for PGC1 alpha, which is the master regulator of mitochondria as well, and related to this. So I think this is one of the best guess that I have.
Yeah, thank you. And Dr. Patricia Joseph has many questions, so maybe she wants to formulate her questions. Patricia. Just open the microphone. Okay, go ahead, Patricia.
We cannot hear you, Patricia.
Now, do you hear me?
Yes, now, yes.
Yeah, now it's okay.
Okay. Hi, I'm calling from Cuernavaca. Hope to see you sometime. Being the mitochondria, see, one of my questions is how do you distinguish some of the changes that could be involved in the cause of the event or as a consequence of cellular activity? And if it's the second, that lies with my second question, will it be another region like PDN or amygdala more active in the high anxiety animals. So to have different regions depending on the characteristic of the animal.
Ah, okay.
See?
You mean on differences?
Yeah, like what you find in Nucleus accumbens on the low anxiety animals, you could be having similar changes in the PDN or in some place in the amygdala on the high anxiety animals due to the stress they suffer.
Thank you. Indeed, yeah, it's difficult to know because we don't know the origin yet. So what we are doing now is to use also genetically modified mice in which we modify Mitofusin 2 to see what's the consequence. I think this answers a little bit your question. If we only have differences in mitochondria, do we have these differences in neuronal structure, in the circuit function, and in the particular behaviors? And this is what we see. When we specifically in the D1, for example, expressing neurons, we downregulate the expression of Mitofusin 2, then the dendritic arborization and the connectivity get poorer, and then the animals also have these alterations in behavior that I was indicating. It depends on whether it's the 1 or the 2. Mice are not rats, but it's very good correspondence with the behaviors that we see in rats. So this is one answer. At least we know that it's cause. Maybe it's also consequence, but at least we know that cause it is because we can manipulate it specifically. And the other answer, we already have some partial, the other question, some partial answers, because I just indicated that when we look at Mitofusin 2 and also mitochondrial respiration in the prefrontal cortex and in the hippocampus, the differences are the opposite. So that the high anxious animals have higher mitochondrial respiration and also levels of Mitofusin 2 in the hippocampus, for example. And now we are trying to understand what's the contribution, to what extent, what is the cause, also what are differences they are mediating there. So understanding better what's the contribution of mitochondria, differences related to anxiety for hippocampal structure, neurogenesis, and also function. So when we looked at the amygdala, we didn't see major differences. That's why I'm not answering so much about the amygdala. But maybe because we don't stress the animals so much. So perhaps if we would stress them, then the high anxious animals would be more prone.
And how about the PDN, the paraventricular nucleus of hypothalamus?
Okay,
that will surely be.
Yes, we didn't look in this model, but we looked in a model of peripubertal stress in mice, and the PVN is showing differences in all these mitochondrial genes. So I think also the animals have a metabolic response that we also think might be regulated by adaptations in mitochondria. But so far, I didn't get any of my lab members to continue with that line of research, but it's very interesting. So
Okay.
we don't know in this model, but in peripubertal stress that induces long-term changes in the animals, in stress, in anxiety-related behaviors, also in sociability, we have differences in the PVN, also in the arcuate nucleus. Not only in the PVN.
In the sorry, can I another little one on what you said about the hippocampus? Will it be the ventral or the dorsal?
It seems that it's both, but because we are looking at neurogenesis, it was easier to see in the dorsal. But it doesn't. It seems that there are differences in both parts.
Well, thank you very much.
You're welcome.
It was a beautiful seminar. Yeah.
Thanks, Patricia.
Now, Dr. Manuel Salas says, thanks for your seminar. And the question is there a role of early maternal leaking on the anxious response of POPs at adolescence and at adult stage? Is there a mitochondrial response during brain development?
Yeah, I wish I knew, because we are relatively new in this field. We didn't cover all these important aspects. I think there should be. I also have the interest of explaining individual differences when, for example, the animals are genetically equal. Where do these differences come from? Of course, they can come from many different components. But one of my hypotheses that I would like to study once is that the early metabolism, not only mitochondria in the brain, but also maybe peripheral one, as it's been shown with manipulations during early postnatal life on the leptin system, change a little bit the individual, the pup, but also change the mother behavior towards the pup, because perhaps the pup is perceived as maybe fitter, so that the mother gets better or more engaged in taking care of the pup, depending on the metabolic state. That's a little bit my thinking, and perhaps all this other behavior, the leaking, etc., plus the metabolic environment or milieu, are very important. Perhaps this is the hypothesis for brain development and making these individual differences in behaviors. But I don't have the evidence, the experimental evidence to answer this.
Okay. The next question. Well, Dr. Manuel Aguilar has several questions, so could you open the microphone for Manuel?
Thank you very much. Congratulations for your seminar. I have a question. The first one is whether do you consider one of the treatments you showed in one of your last slides could be considered better or more promising than the others, or maybe a combination of these could work?
Mm-hmm. Yeah, we've been doing these studies a little bit opportunistic and not coming from always an a priori reason. We did, for example, the medium chain triglycerides because indeed if there is an energy deficit, these medium chain triglycerides produce very quickly ketone bodies that are very prone to cross the blood-brain barrier. So one gets very quickly a huge, very, I think, not useful, but efficient source of energy in the brain, because they can produce much more energy and ATP than glucose, and also they are less toxic. They produce less ROS, etc. Eventually, they were amazing because within 1 week, the high anxiety of the animals was totally reversed. They behaved as if they were not anxious and they were highly confident, etc. But we did this also in collaboration with Nestlé. And when we stopped this study, we didn't continue because this is the problem sometimes in science, that one needs funding to do particular studies. So when the funding was finished, we didn't continue. Then there was opportunistically also the possibility to test other compounds. And that's the way we've been doing until now. Now we are changing a little bit because indeed now we are gathering more information also from the spectroscopy to tell us, and also from preclinical work in animals, to tell us which maybe pathways are the most interesting to target. And that's why now indeed we have a different approach. And the problem is that indeed I also believe that perhaps a combination of treatments could be better than just targeting one of the pathways. But this, again, to do the research is highly time consuming because one needs so many groups to study everything. So the answer is indeed, there are a lot of promising possibilities and interesting ones, but I am a bit frustrated that we have to advance so slowly because even in humans now, to just do a study, we required almost until now, I think 9 months, preparing the ethical license to be able to obtain the permit to do the studies. So one cannot try or test so many things. But I think, yeah, slowly and getting more information also, we will be able to have a better, a more rational approach to these treatment studies.
Oh, thank you. Okay, and since this problem is related to stress, do you think that proper management of stress alone or in combination with one or more of these treatments in individuals with this high anxiety trait could prevent the development of the stress-induced depression?
Yes, I think so. Some people are now self-medicating themselves because they know that some of these nutrients are available. You can buy them easily through Amazon, like taurine or N-acetylcysteine or many of these different treatments that are good for the functioning of mitochondria. I think I know some people that are taking them and I see some effects. I didn't try it myself.
Okay, that's good news. So, thank you very much and congratulations again.
Thanks so much.
Now the next question is by Marisol Orozco, and it seems that only the modulation of the electron transport chain is relevant for anxiety. Have you explored the involvement of any other mitochondrial metabolic pathway? Not only electron transport, but something else.
Because I think what is important here is that it's true. Eventually, what we did had an impact in anxiety, but the view of what we've been doing and what we do is that I don't know where anxiety comes from or wherever, but anxiety has many different manifestations. We are anxious, and then maybe indeed we get sympathetic activation with all the symptoms that we know from anxiety and stress. And then we have cognitive ways of thinking that anxiety is targeting maybe the prefrontal cortex for reasoning, etc. And another manifestation of anxiety that perhaps is different in different anxious individuals is indeed to make us cautious. No? If there is a threat, instead of energizing us, is to make us retracting and have this passive copying, etc., keeping the energy. So that's why what I show until now, I think, is to clarify. I think it's how anxiety affects the motivational system, that eventually, of course, everything is in a loop. If we have a motivational system that it's self-confident and that feels the energy to go out in the world and deal with threats, this is going to somehow, we don't know how, but I think it would be interesting to study how this increase on this type of self-confidence or this boosting in energy kind of reflects back in anxiety and affects the anxious individual, that it's more confident and then can cope better with things. I think I just went into another type of response, but I think it's important to clarify that I'm not so much relating everything only with anxieties, how the anxiety system taps on this motivational system. But indeed, I'm thinking what we were targeting. Well, the Mitofusin 2 is not targeting only the electrotransport chain, because what it's targeting is the fusion of the protein, the capacity of the mitochondria to respond dynamically to changes in demand of activity. So in this case, this is not the electrotransport chain, and we see that it has a huge impact. Also, we are now looking in more detail at how not only this Mitofusin 2, but these interactions between the mitochondria and the endoplasmic reticulum are working. Because indeed we see that there are differences, and it seems that this is critical for how these circuits are working. So now we are manipulating these interactions between the mitochondria and the endoplasmic reticulum with different types of tools that allow us to put them together with linkers, or to measure how many proteins are there, if there is a different protein composition in these MAMs that are called the mitochondria linkage with endoplasmic reticulum, this type of addition molecules. One can explore if they are different or not. So the answer is indeed, until now we covered the electron transport chain. We cover this. We explored once also the ROS. We gave only once, I have to say, we didn't try different treatment schedules or also doses. It was with one drug that targets the ROS system, the antioxidant system, and this didn't have an effect. Now we have a study also with a company in which targeting the ROS system will see also effects in motivation. So yeah, this is the answer, but we didn't publish it yet. But it's not only the electrotransport chain. I think eventually in the mitochondria, everything works together. So I think maybe we measure different aspects, but eventually it's just like when we look at the brain, we are looking at different levels or different techniques, but every mitochondria works together. And if there is an alteration, perhaps in how the calcium is buffered because of the interactions with the endoplasmic reticulum, eventually this is going to affect also the electrotransport chain. So I think there are many different pathways. To what extent they work independently or we are seeing different manifestations of a phenotype, I don't know. Yes, thank you.
Okay, now the next question is by Dr. Roberto Prado. And he says, you have shown that mitochondrial activity correlates with low and high anxiety states. Does mitochondrial activity correlates with learning and memory capacity?
Yeah, it's very interesting, but we couldn't do these experiments yet. But perhaps because we know that when we classify animals for high and low anxiety, the same rats, we know that particularly with learning, high anxious animals, for example, when we put them in the Morris water maze, they take longer to learn. So I don't know if mitochondria is in this case responsible, but indeed there are differences. And this is work that we did several years ago, and that was, we found a relationship also not with the glucocorticoid receptor in that case, but with the mineralocorticoid receptor. So I think there is very interesting work there because it makes all the sense that there is a regulation. Thank you.
Okay, and Rogelio Pegueros asked, is the stress impact on mitochondrial activity the same in neuron and glial cells?
Yeah, until now, we were not looking so much at glia. It's in stress. We've done also gene sequencing for the nucleus accumbens and the prefrontal cortex after chronic stress, after I think it was a new type of stress that we developed in the lab with multi-sensory approaches or stressors, and then for also chronic unpredictable. And there we saw in the chunk of tissue, because we were not differentiating, differences that were stronger in the prefrontal cortex. What we see until now in the astrocytes, it's just comparing high anxious and low anxious, but we can also consider our high anxious because they are in the stream as perhaps chronically stressed, because probably these animals are always having a lower threshold of responding as stressed to any challenge. And in this case, we were not looking at astrocytes, I have to say, now for a couple of years, because when we looked at the astrocytic fraction at the beginning, we didn't see differences in the respiration of mitochondria. But now that we look at the electron microscope images, we see, as I was indicating, differences also in the contact of the mitochondria with the ER, and also in the location of mitochondria in the astrocytes. So we started looking at astrocytes, and this is ongoing work. I don't have any more details, but I think perhaps, and that's why I'm really interested in looking more into the details, they should be critical in all this kind of complexity, because when we do spectroscopy and we just look at metabolites, astrocytic metabolites come as critically different between dominant, subordinate animals, and also how they react to the chronic social defeat stress, for example. And there we had differences in alanine and lactate, and these are two astrocytic metabolites. So I think it's all open because we know very little about all these fields. So I think there are very, very many questions that still we need to answer. Thank you very much.
Yeah, and I have the last question. Is there a correlation between shape of the mitochondria and function? Because you mentioned that the high anxiety has like a bigger mitochondria, elongated shape. And then how this correlates with the functions that you see restored when Mitofusin 2 is replaced?
Exactly. I don't know beyond a little bit of an abnormality because the mitochondria shape of the high anxious animals looks to the expert eye a little bit swollen. So I don't know if when there is no deficits, the shape would correlate. We are looking now a little bit at analyzing a lot of mitochondria in different dendrites on dendritic parts, also distance from synapse, to see if the shape and also the number of mitochondria has any type of correlation with these parameters. So yeah, just to understand better, yeah, how mitochondria refers to a synaptic compartmentalization, or maybe even the size of synapse, etc. We really don't know. Here, when we go to the extremes and then the mitochondria becomes swollen and maybe a bit abnormal, indeed we see differences. And what would be very interesting now is to use, for example, mitoTAC approaches that it's going to allow us to precipitate the mitochondria from specific neurons or projections, and then do proteomics, for example, to understand what could be different in this shape. Yes.
Okay, thank you.
A lot to do.
Yes.
Yes.
Yes, well, it's the innovative of the work. So, Dr. Isabel Mendez says, thank you for your seminar. And I also take the opportunity to thank you for this wonderful seminar, very provocative work, and amazing work that you are doing now. So thank you, Carmen, for your presence here. Hope it will be physical in the coming years, but we still don't know when. So
thanks so much.
Somebody wants to do another question. Gina? Gina sends you a hug, a big hug.
Thank you.
Okay.
Thanks so much. And also for the discussion, it was very stimulating, and happy to see you all, or the pictures of some of you.
There's one more question for my student. Is there time or no?
Okay.
Yeah, you can. He wrote this: if the stress impact on mitochondrial activity is the same in neuron than in glial cells, do you know something about it?
No, we have to do all these studies. We don't know yet. We don't know.
Yeah.
I think it would be very good to be able to fact sort these different cell types. Another way that we used now is this Cartana, which allows you to get a lot of gene expression in many different cell types, but it's not high throughput at all. So I think, but yeah, I think.
Because there's always glucocorticoid receptors in the glial cell, so
Exactly.
can be. Yeah.
Mm-hmm.
Thank you, thank you. Muchas gracias. Un abrazo.
Muchas gracias. Que sigáis muy bien.
Sí, gracias.
All right. Well, thank you very much for your seminar. And Maru, if you can stop the transmission.